Auzubillahiminash-shaytanir-rajim. Bismillahir-rahmanir-rahim. Dear students of 12th class, subject Botany, Assalamu alaikum wa rahmatullahi wa barakatuhu. Dear students, you know we have discussed many questions so far in the first unit, and today we will discuss or we will throw the light upon one more question, that is the pollen-pistil interaction. Dear students, we have studied the pollen grains and we have also thrown light upon the structure of a pollen grain, and we have come to know that it consists of two layers. The outer layer is called the axine and the inner is called the intine. And we have also seen that the pollen grains germinate on stigma of the style of a carpel. And here in our today's topic, the question is pollen-pistil interaction. Pistil here means carpel, and upon the carpel we see the stigma on which the pollen grain germinate and bring about pollination. So you should not be confused here. What is this pistil? It is simply the carpel, and it is now in our question pollen-pistil interaction. So we will see here how the pollen-pistil interaction takes place. Dear students, pollen-pistil interaction involves the provision of favorable physical and chemical conditions for proper germination of pollen grains on stigma and styles of carpels of female flowers after the pollen grains are transferred from anthers to stigma by the act of pollination. Dear students, you should remember here that the pollen-pistil interaction happens only when there are the favorable physical and chemical factors, physical and chemical conditions, and that will govern the proper germination of pollen grains on stigma. So the physical and chemical conditions for proper germination of pollen grains on the stigma and styles of carpels of female flowers is important when the pollen grains are transferred from anthers to stigma by the act of pollination. So what happens? The axine of angiospermic flowers is composed of a polymer called saporopollenin. The axine, as I have told you, it is the outer layer of this pollen grain of angiospermic flowers. Here it is composed of a polymer that is called the saporopollenin. The cavities present in the axine are filled with specific proteins, enzymes, glycoproteins, lipids, etc., which are derived from tapetum during saporogenesis. This axine of angiospermic flowers have the polymer saporopollenin, and the cavities present in this axine, those are filled with specific proteins, enzymes, glycoproteins, lipids, etc., and those are derived from the tapetum of the anther and during the saporogenesis. The chemicals act as recognition factors. These chemicals, like the proteins, enzymes, glycoproteins, these act as recognition factors. They recognize how the germination can take place by the pollen grain on the stigma. The stigma of flowers also possess, the stigma also contains these chemicals like proteins, lipids, amino acids in the form of free-floating secretions. So we should remember here that both the axine of pollen grain and the stigma of flower contains these chemicals, or the cavities of these are filled with these specific enzymes, glycoproteins, lipids, etc., and they act as the recognition factors for both pollen grain and the stigma. Now, during pollination, there is interaction. So what happens when the pollen grains are transferred from anther to stigma? and when the pollen grain stays on the stigma, there is interaction between these chemicals of both the pollen grain and the stigma. And these secretions of pollen grains and stigma, which determines, and it then determines these chemicals now determine whether pollen grain can germinate on the surface of stigma, or it can and can cause fertilization, or it is rejected. Fertilization means here, can it give out the pollen tube, pollen tube. So these secretions of pollen grains and stigma, they can determine whether the pollen grain can germinate on the surface of stigma and can cause fertilization, or it can be rejected. So now you will come to know that how the pollen pistil interaction takes place. This is nothing but, here you know, and you can remember that the pollen grains contain chemicals of different quantities, different kinds, and the pistil also contains these things, and they can act as recognition factors and can brought about the fertilization, or it can be fertilization can be rejected. So this was all about today's topic, today's lecture, pollen pistil interaction. I hope you have understood now the pollen pistil interaction. Stay home, stay safe. Thank you very much.